fourth podcast in our information domain. In this one, we are going to spend some time with viruses. So we are going to look at answering two big things, what they are and how they work. So first off, what they are. Okay, they are an obligate intracellular parasite. That means they can't survive without something else alive to live in. So just out and about by themselves, not going to work. But they are made of really two things, two molecules, genetic information, whether that's DNA or RNA, and a protein coat that we refer to as a capsid. And so, yep, that's what they are. So here are some different types that you can have, and just some various types of viruses. There are many of them. They all have different interesting shapes. So the tobacco mosaic virus is a retrovirus with RNA inside of it. That capsid around it kind of creates this rod-looking shape. Adenoviruses, are there with their again, the capsid DNA inside. Your flu virus, your influenza, same idea. You're going to have a capsid with glycoproteins that have sort of, those are basically like little receptors that are going to be able to recognize host cells. And then you have things like bacteriophages that to me always look like a landing module for Mars, where they have their protein, and then they have the DNA on the inside. Bacteriophage, that is what Hershey and Chase used when they did their very, very famous experiment. All right, so since viruses can't carry their life cycle out independent of a host cell, they're not cells. They're not traditionally considered to be alive. And for some people, this matters. I'm not sure why, but it does, because we know that they just cause and wreak lots of havoc on us and other organisms, regardless of whether they're alive or not. It's a thing. They were first discovered by Dmitry Ivanovsky, who was studying how tobacco mosaic disease transferred. And what was discovered was that there was something smaller than the cell that was actually the root cause of the disease. And so it was pretty nasty because it will wipe out tobacco plants. On a personal note, like it matters, but hey, people care. So this is what ended up happening. So basically extracted sap from the plant, got passed through a filter known to trap bacteria, but then you rub that back on the plant, and then the healthy plant became infected. So we knew it was something smaller than any kind of cell that was causing this, and that's the deal with viruses. They tend to be extraordinarily creepy, tiny buggers. So since then, viruses, for all domains of life, there is if you're a critter, there will be a virus that will infect you, and most are harmless, and we can live with and be fine with for your entire life and maybe not even know you have it or know you have it on occasion. And some are quite deadly, and which we know as we tend to battle them. Herpes virus, which causes cold sores and other inconvenient irritations elsewhere, is one you can live with, but it is easily passed. Then you have things like measles. We've recently had the sudden outbreak of measles again, even though we've had vaccines for it. Smallpox, polio, these are all viral. And then you have the flu virus that we try to combat every year, and with hopes of not having any sorts of massive pandemics like we did in 1918 when it killed tons of people. So, a little cold sore look. Again, measles, polio, smallpox, and then you have the influenza, and then things like H1N1, and various other viruses that you hear about. And of course, then there's HIV, which we'll come back to. There are viruses, like I said, all domains of life have a virus for them. So there's prokaryotic viruses. These are known as phages, and it comes from this idea of a phage, like to eat, like a macrophage or anything like that. Phagocytosis, right? That engulfing means eating. So their habit of eating wads of bacteria in a petri dish is where the term comes from. And it's considered, maybe thought that there's 10 to the 31st phages on the planet. And if you're not sure, that's a really big number. Really big number. So just saying. So a larger number than the stars in the universe, okay, just to throw that out there. If you took every phage, as teeny tiny as they are, and lined them up, that's roughly 100 million light years long. So wrap that one around your head. several billion times. So here is a T4 phage that's infecting E. coli. This is a phage that we, see. Notice the size. We've got 0.5 millimeters is terms of that. We're talking teeny here. So that size, 500 micrometers. Infection plaques on a bacterial one where it's been broken down. You have two types of phages. You've got virulent, which has a lytic cycle. Virulent means this is classic. It's going to infect, go in, bust open the cell, kill the cell in order to continue its life cycle around. So you can see it sends in its genetic material. The cell becomes a viral factory. You bust open all of those and we reassemble as they're being all assembled. Then new phage leaves to go infect other cells. Then you have temperate phages, this lysogenic life cycle. The phage DNA will splice into the bacterial chromosome. Then when it replicates the DNA is also replicated, and you can have lytic, as needed, you can make it. So it's not constantly made and then busted out. It can only go when it's necessary into its lytic cycle. It can kind of just remain in the genome until necessary. So lambda phage will do this. And lambda DNA is what we looked at our restriction simulation. So here's kind of how that works. You get the infection, incorporation into the genome, cell divides, it'll kind of hold on to that genome. This is the lysogenic cycle, and then as soon as it enters the lytic cycle, we're actually going to pull that DNA out and it's going to become that factory and then lyse out all of those cells. In some cases, we can use phages as a way to treat bacterial diseases, because since they're very specific, like all viruses are extraordinarily specific to a single host cell, we can do that. So it's been used as a thing, but we're trying to incorporate it more so as a way to combat different types of bacterial. Maybe this could be an answer to things like antibiotic resistance bacteria that grows and infects people. Perhaps we could use that as a thing. Wouldn't that be cool? And then there's eukaryotic viruses. As we know, every critter has a virus. Very, very diverse, a lot more range than a phage. DNA or RNA genomes, so you have sort of regular viruses and what we call retroviruses, either single or double-stranded. Many also contain, along with their protein coat, a lipid envelope that surrounds it. So you can see sort of the difference between a DNA virus here and an RNA virus, so you can kind of look at how the process differs from the way that they enter a host cell. In a DNA virus, you will not have the actual piece won't actually totally enter the whole cell. You're going to get parts of it that will enter. And then the same thing with a retrovirus. The capsid enters and creates more. So take a second, kind of pause and kind of look at the different pieces of how this works. So here's some kind of classes that we can have. You have various types of viruses. You get double-stranded DNA viruses, adenoviruses. Those are normally respiratory type viruses. Papavirus, papillomavirus. These are warts, cervical cancers. So like when you hear about HPV vaccines, that's that one. You've got herpes virus. You got simplex 1 and 2. Different types that cause shingles and shingles and chickenpox are part of this class. And then you've got like smallpox, and then single-stranded types, as well as double-stranded RNA types, which are your retroviruses. So yeah, so just different types. Here are some other ones, some single-stranded RNA ones as well. You can kind of look through. We're not going to, in terms of generalities, I'm not going to get you super specific on the types, but I want you to know the general types of viruses that exist. And so kind of look through, you can see here is our Ebola. It's a single-stranded RNA that's a template directly for messenger RNA synthesis. And then you have single-stranded RNA, which is a template for DNA synthesis. So what this retrovirus does is it, like HIV, will take that single-stranded RNA, reverse transcribe it into DNA, then you've got that genome incorporated in, and that's something like what HIV does. So here are a couple examples of some of those animal viruses, and this is kind of where we'll kind of call it. So you've got your influenza. It's an RNA virus, easily transmitted in aerosol form, so they can survive outside for a little bit. You know, and so this RNA virus enters in. You can see kind of how it goes in, enters into the cell. Information gets sent into the nucleus and kind of enters into the nucleus, not the whole thing, just the genome will enter into the nucleus. That's a little bit different, right, than a prokaryote. So the virus comes into the cell, but not the whole virus is going to enter into the nucleus. Only the genetic information enters into the nucleus. You're going to get some transcription splicing, some mRNAs being created, and then those head out to the ribosomes to ultimately help to create the new viral pieces. We all know flu symptoms, right? It's all respiratory primarily. You're going to get fever, achy. What you you will kind of get stomach sick, but the key is kind of having that respiratory piece as well. Flu is responsible for more deaths than any other known virus, even HIV at this point. While we have, what's amounted to be 40+ million people infected with HIV, as far as deaths go, flu is the #1. And it mutates very rapidly. This is why it's so important as we try to figure out vaccines every year. They have to basically guess. They guess what is going to be the most prevalent type of flu based on the mutation rates to try to guess the vaccine that they're going to create. So that's why it's kind of a crapshoot, honestly. And you can have 2 strains that can kind of have additive effects if you get 2 flu strains and have a huge large-scale viral recombination, and it just gets messy. So over time, Influenza A, we have different subtypes in the population that we've numbered over time. The big one of 1918 was H1N1, and kind of where it's come, how it's returned, what other ones have been around, where do we go from here? It's, again, that question that's out there. So naming for flu viruses there's an actual convention, just like naming living things. So when we name viruses, you have the virus type, geographic origin, the strain number, the year of isolation, and that's the breakdown of how you get it. So when you kind of go down, and then the subtype of that. Okay, so that's just how you name them. So here is a little YouTube. So directly go to the Prezi if you want to watch this. It kind of showcases sort of the idea through some 3D animation, what's going on with flu virus, so it's kind of cool, so definitely spend some time there. The other big one that I'll take a minute to talk about is HIV. We have known about HIV since the early 80s. Very, very gnarly disease and terrible disease, and has influences and affects us worldwide. So at this point in your life, if you don't personally know somebody with HIV, I hope you never have to go through that. I've actually known somebody with it and who has passed away from it, and it's pretty rugged. So it's good to learn about it. It is a retrovirus. It's RNA genome. It does have a code that does reverse transcriptase. So the transcription makes the DNA copy, which then splices into our host cell, specifically our T4 lymphocytes. And it is that backwards RNA to DNA. So right off the bat, this little thing violates central dogma because of that. Here's the way it works. It works its way by attaching to very specific receptors on the host cell. These little receptors come in and open up, become part of the cell membrane, and throw this basically the actual capsid with the RNA into the cell, where it falls apart, and then that viral RNA will then join into the nucleus. So here's kind of how it works. Entering our helper T cells, which are those lymphocytes that we mentioned. Very, very specific. This is the cell it infects in humans. It won't infect any others, but it's an important one because this type of cell in our immune system acts as like our scouting mechanism, and it's the scout. It's what we send out to look and signal if there's anything in us that's not supposed to be. And if we lose our scout, we lose our ability to recognize. And that's why people who pass away from ultimately AIDS, it's not from the virus, it's from the diseases they get because they weren't able to fight them off. So that's how it works. And you get acute tons of infections that are very, very flu-like in terms of what you get, in terms of the infections that you get. So numbers that are very specific but very similar to flu. And it ultimately breaks it down and breaks down your immune system. And once you get to a point where your body no longer has that ability to, to fight off things day to day, you move from being HIV positive into having full-blown acute immunodeficiency syndrome, which is AIDS. So here you have it. So this is kind of how they look at the lymphocyte count, what happens as things change in terms of the infection versus the number of cells that you have. Okay, and how the HIV RNA copies in your cell, basically in your plasma, showcase. That's how they can actually track. So you can see as that increases, the number of cells that you have decreases, and you're going to be in trouble during that point. So only through bodily fluid exchange can HIV be can be transferred. It can only live outside the body for like 20 minutes if it's airborne or in any kind of liquid. It can't survive very long, so it's direct liquid transfer. And you can see just sort of the spread of where you can see it is most prevalent. You find it in sub-Saharan Africa in terms of its most prevalent areas, to the point where a number of years ago, I remember reading, maybe 10, 15 years ago now, I remember reading how it was going to be a big deal that Sesame Street was going to introduce into South Africa, into their group of critters on the Muppets, right, on Sesame Street. They were going to have one that was HIV positive because the numbers were as close to being 1 out of 3 people in those particular areas were HIV positive, and felt the need for educationally to actually get that out there. So there was going to be an HIV positive Muppet on Sesame Street. So lots of treatments. So how do you treat this since it can't be cured? And you do it with drug cocktails. You try to keep your internal environment constantly different so that you don't get the ones that have particular mutations growing, so you're constantly changing and constantly shifting your drug cocktail, okay? And there are people who are going to be more prone to survive longer with it, less. You look at somebody like Magic Johnson, who I think, oh man, 1992 or 3 maybe, was when, it came out that he was HIV positive. He's still going. And then there are people, depending on the strain, that might not make it a year. So it varies so much in what the chances are. So anyway, learn some stuff about it. December 1st every year is always known as World AIDS Day. So spend some time. Learn a little bit about the virus, what's going on in the world, how it's treated, how we get education out about it, and how we can stop it from being spread. Super important. Unless we find a cure, which we'll see. But even still, we can stop it. It's preventable. So plant viruses, there's your other one, right? And so plants get their viruses too. Believe it or not, they can usually end up with these kind of crazy blotchy pigment patterns that result from their viruses. So again, we saw earlier the tobacco mosaic virus, because it creates these splotches, gave it a very mosaic-y looking thing. So there's other ones that can be out there too to cause these problems. And lastly, okay, so viruses themselves, they're not just the most simple infectious particles known. I'm not going to say, well, you can call it a critter, whatever you want to call it, whether it's alive or not. It's not the simplest. There's actually other ones. So I'm going to just kind of cover this real quick. There are viroids. This is disease-causing RNA in plants. And they don't code for an actual protein. So you've got sort of this potato viroid here that you can see is an oddly circular piece, or not kind of circular, double-stranded piece of RNA that we'll actually call it. And we call these a viroid because they're not viruses, they're not a full-fledged virus, and they're just those pieces that cause problems. And then we have prions. You guys have heard of these before. These are rogue protein molecules that are extraordinarily stable, and they're going to cause other types of disease. They're not genetic. No genetic material at all, which is wacky weird when they were first discovered. These are what cause, prion diseases are what cause things like BSE, bovine spongiform encephalopathy, that's your mad cow, CJD in humans, Creutzfeldt-Jakob disease, basically the version of BSE in humans, and scrapie is the version in sheep, and then you have chronic wasting disease in deer. These are some examples of that, and they all ultimately do similar things to all the individuals, but these particular proteins vary in shape a little bit, and they're a little bit different. However, because of scrapie first shifted to cows, and then cows to us, so these alterations of these prions, and if they're there, what they'll do is they attach onto a good protein, and the shape changes. And when that shape changes, oh boy, look out. So that spongiform plaque that forms in brain tissue, you get these big plaque buildups, and of course that kills your brain tissue, and that's no good either. So there you have it. We have what viruses are, and we have what viruses aren't. So that's a virus. And so from this, make sure that you can do a couple of these, that these are the things that you can do and that you can compare. And so we figured it would be, we can't talk about information and how things pass without bringing these guys up. So that's where you have it. And then from here, we're going to start talking about how cells make more cells. So that's where we're going next. So have a great night, and we'll talk to you guys later.